Hello and welcome to the World Advanced Therapies and Regenerative Medicine Congress Interview Zone. My name is Freya Leask, I'm the editor for RegMedNet and I'll be your host for today. And I'm very pleased to be joined by Fred Chereau from LogicBio. Fred, thanks for joining me.
Pleasure.
So for those of us that may not be familiar with LogicBio, can you tell us a little bit about the company?
Sure. So LogicBio is a genome editing company developing a technology which has been initially discovered at Stanford in the lab of Mark Kay, who is one of the key opinion leaders in the gene therapy for many, many years. And basically we are focusing on developing treatment for rare disease and especially rare disease for pediatric patients, where you want to intervene very early on in the life of the patients before some non-reversible sequelae occur.
Interesting. And how did you yourself get involved in LogicBio?
That's a good question. Actually, I've always been passionate with regenerative medicine, and I spent a lot of my time at Genzyme, where I was in charge of multiple programs in cell therapy and gene therapy with a different type of gene therapy at the time. It was in 2000, 2005. And I've always followed the innovation there. And when it was time to find a new opportunity, about 3 years ago actually, I got interested in the technology developed by Mark and two of his former postdocs. One of them is now a professor in Tel Aviv University and the other one is in Australia, so we are very global. And I found this technology was fascinating because very different than what we call canonical gene therapy, which is basically the gene therapy with an episomal expression that most of the companies are doing. And we are doing editing, but we claim we are doing genome editing because we are very different than the other companies you may know who are developing editing with nuclease-based.
Okay, so LogicBio has a platform called GeneRide, is that correct? So what is GeneRide? Why is it so much better than everything else?
So much better. we still have many things to prove, but no, GeneRide is a promoterless, nuclease-free genome editing platform. So why is it important? And coming back to what I was saying, if you look at the gene therapy or promoter-driven gene therapy, I think it's a fascinating technology. And as you know, we have now some product on the market, and very soon we will have some others. There are a couple of pitfalls with this type of technology. It's not a great technology to go after cells which are dividing, So, naturally, you can imagine it's not a great technology to go to some cells, especially in children, when the children are growing and their cells are dividing at a pretty high rate. So, another, potential challenge we see also is the use of a promoter. It's very fortunate that nothing happened in clinical trials to date, but even if people called AAVs non-integrative vectors a few years ago, we know now that they do integrate. They don't integrate at the same rate as others, but they do integrate. And we think that if you could not use a promoter, it would make your product a little bit safer. So to defeat this protein expression, the loss of protein expression you have when the cells are dividing, you need to edit the genome. You cannot stay just episomal. And to edit the genome, you have different ways. You can use nucleases, but you can also use homologous recombination. Homologous recombination is a naturally occurring process, which is basically what your cells and my cells are doing now to repair minor DNA defects. And so that's what we have decided to enhance, to insert in a very specific place of the genome, the gene which might be missing. So we think with not using nucleases is interesting because nucleases are very potent very potent nuclear scissors, or not nuclear, molecular scissors, and it's great. But in the case of CRISPR, for example, the GPS guide, which is determining where this scissor should cut and insert, is pretty short. It's only 20 bases. The problem it could be today, it's a lack of specificity. The likelihood that you find the same bases, 20 bases, somewhere else on the miles of genomes we all have in our cells is not new. In the case of GeneRide, our GPS system, we call that homology arms, are between 500 and 1500 bases. So the likelihood to find the same sequence somewhere else in the genome is very limited. I mean, in biology, I don't think you can ever say never, but I think it's very, very limited. So we think that by not using promoters and not using nucleases, we can develop a very efficient and long-term product which should have a very good safety profile. So we believe it's an ideal product to target these pediatric diseases where, once again, you want to intervene before some non-reversible sequelae occurs in these patients.
And does a platform like this reduce the risk of off-target effects as well, as it is so specific?
So if you have multiple types here of target. First, you have the off-target, which is driven by the AAVs. And every AAV company, as I was saying before, every AAV technology, as I was saying before, will have this type of target. We will too. But because we don't carry a promoter, if in case of uncontrolled off-target, we insert next to an oncogenic site, it should be inert. You know, we should not trigger carcinogenicity. The other type of target which could happen is a little bit what I was describing with the nucleases, where you cut or you insert in places where you don't want to do. And once again, with this 500 to 1500 base pair guides, the likelihood to be off target, directed off target like that is very limited. And the last thing is in the liver, for example, we use albumin as the gene we target. We target the very, very end of the albumin to take advantage of the albumin promoter, which is very potent. And albumin is not expressed in any other tissues. I mean, maybe a little bit in a couple of other tissues, but mainly in the liver. So we think that even if there is some off-target integration in the albumin locus in some other tissues, it should not create any protein production.
Okay, so let's talk about applications of this technique. So LogicBio recently received orphan drug designation for LB001. Congratulations for that, which is indicated for methylmalonic acidemia.
MMA.
So stick with MMA, yeah.
Right.
So what is MMA, and what is LB001? How does it target MMA?
So MMA is a rare organic acidemia, and which happens at birth. I mean, the manifestation of the disease can happen at birth, so very early. And basically, the children are missing a gene called MUT, M-U-T, which code for an enzyme which helps them to metabolize proteins. And if you don't give them proteins, they don't grow. If you give them proteins, you start a storm of metabolic unbalance, you know, which can lead to coma, CNS disorders, and non-reversible CNS disorders, and even death. So there is no treatment for this disease today. There are about 2,000 patients in the US, about 3,000 patients in the Western world, I mean outside of the US. And the only treatment or only tool the physician can use is a very low protein diet and high calorie. So these children, and as they grow also, the only thing they can take as food is formulas, and that they have to take very precisely.
Wow.
And sometimes they can use them, I mean, ingest them. Sometimes they have a GI tube, and ultimately, I mean, the last thing we can propose these patients today, it's a liver transplantation at some point.
Okay, and obviously LB001 is a gene-edited therapy. What does it target to treat MMA?
Well, it's the same as the rest of the platform, and maybe I can tell you a word about the rest of the platform. But basically, with GeneRide, we can insert where we want by homologous recombination. So I said, in the case of the liver, we have decided to insert at the very end of the albumin locus to take advantage of the promoter of the albumin locus. And with for MMA, basically we are inserting the MUT gene at the very end of the albumin locus, and we express the MUT, which codes for an enzyme, which is going to help for the metabolizing these proteins. We have other proof of concept, and actually the first proof of concept the company has developed was in hemophilia B. It was the same type of technology. We were using still albumin. We were inserting factor 9 in this case at the end of the albumin, and then the mice started to produce factor 9. And that's, it tells a lot about the modularity of the platform. We have published, I think, 4 proof of concept in hemophilia B, alpha-1 antitrypsin, MMA, and Krieger-Najjar. And there is another one we have not published yet, but we will publish soon. And we have many others ongoing in the company. And the modularity of the platform is such that when we still stay in the liver and still target the albumin, within the overall vector, the only thing we have to change is the gene. The rest stays the same. And everything we have learned in program 1 can be used in program 2, 3, and 4. And so we are very hopeful that, as soon as we would have made a human proof of concept in MMA, we will be able to roll over to other indications relatively quickly.
Fantastic. Okay, so let's move on to what you're talking about at the World Advanced Therapies meeting, which is latest methods and protocols. Obviously, science is moving rapidly forward all the time. There's new techniques, new findings to do protocols better. But then there's the consideration that regulators like us to confirm those protocols and have them consistent and robust from, all of the clinical stages up to regulation. How can developers know when the technology is ready to integrate into their protocols? How can they balance that dichotomy?
I would say that the billion-dollar question, and it's not a question only for regenerative medicine. Science is going faster and faster. We have better and better tools to help science being developed faster and faster. And I think, it's at some point you have to decide when you think your product, your platform is ready for clinical trial. Obviously, it goes after years of development, sometimes in academia, but in vitro first, in vivo, and you test in multiple animal models. And when you think you have something which is optimized and safe, which is, I think, the most important thing, you decide to go to the clinic. Now, and as I'm sure you have noticed in regenerative medicine, you have a new publication almost every day, which is kind of outdating the others. If we try to optimize the platform as the science is going, I think we will never go to the clinic, And if we never go to the clinic, we won't be able to really validate any of this technology, because what we learn in animals is very interesting, it's key, but it's not what we are aiming to do. And obviously we want to treat patients. Some of these patients are in situations which are terrible, and sometimes their life expectancy is limited. It's always a balance in between deciding when it's the right time and trying to deliver a good product to the patients.
Fantastic. So finally, what can we look forward to from LogicBio in the next few years?
It's also a good question. I hope that in a few years we will have one and maybe more than one proof of concept in humans. We will have many other development targeting the liver, because there are many diseases where we think we can go. We may have coming back to your previous question, actually, GenRI 2.0, which will help us to target another type of disease we cannot target very well today, because every technology has its limitation. And what I hope a lot too is we will have demonstrated, and hopefully it's going to be in a short amount of time, that we can go to other tissues, like muscles or maybe CNS, where there are many diseases which could benefit from GenRI. Obviously not targeting the albumin, because that's not expressed in these tissues, but targeting other genes and taking advantage of these genes to take a ride on the genes. That's the reason why the technology is called GenRI, and expand like that the array of indications we can target. And I think, hopefully, LogicBio could become one of the leaders in developing genetic medicine for rare pediatric diseases.
Mm-hmm. Well, I've got my fingers crossed for you.
Thank you very much.
Fred, thank you very much for joining me.
Thank you.
And thank you all for joining us. And I'll be back with another video from the interview zone very shortly.